Welcome to this research project that is hosted here at the Laudato Si Research Institute, which is based at Campion Hall, University of Oxford. This research project is exploring the new scientific development of gene drives. We are exploring at least three key questions. One is, what is a gene drive? Secondly, what are the implications for policymaking and public engagement? And thirdly, who does it most impact? How can we engage and understand what the implications for real people?
Well, at the heart of the project, there's a real-world problem. The threat of invasive species is enormous. They affect animals, they affect people, they affect ecosystems, they indirectly affect food security because the animals are lost, and they cause $4 billion annually of losses. It's a huge problem, quite apart from the inordinate suffering that they cause, because some of these invasive species, such as the screwworm, for example, they're flesh-eating insects. There have been various techniques that people have used to try and control the screwworm, knowing both the economic loss and the huge suffering that's taking place. But unfortunately, none of these techniques are very successful. The screwworm persists, and of course the screwworm doesn't respect geographical boundaries. It can spread to other countries and nations, and therefore carry on in its path, leading to suffering and more economic loss and so on and so forth.
So over the last 20 years, researchers have been playing with the idea of, can we create or recreate natural gene drives in a synthetic way? And that might be useful, because then we can spread a trait through a population that might be useful for humans. Gene drives occur in nature, and they're found with many different mechanisms and incarnations. So gene drives would allow us to very efficiently address some of the largest public health and conservation and agricultural challenges that we're facing throughout the world. And gene drives can make existing methods much more efficient than they already are. There are potential ways where you can build in safety mechanisms into the research you're doing. I would say the majority of research into CRISPR gene drives have used exploited a fact about the CRISPR nuclease, where you can take one component out and then put that somewhere else in the genome. And as long as that is present, it behaves like the full gene drive, but it would never spread because the thing you've removed becomes limiting very quickly. But that still allows us to do the research in a safe manner.
Our research project is particularly interested in the ethical, social and wider political issues and public issues around invasive species, with particular reference to the screwworm in South American countries and some of the social and political and cultural issues in how this may be received or not in some parts of the world. We are also, of course, involved in thinking through some of these issues with the Catholic ecclesial leaders in Rome and elsewhere and locally, but because these are Catholic majority countries, so there are serious ethical and theological questions around how we should use our technologies.
New biotechnologies enable us to think in fresh ways about problems that need addressing, but at the same time they require clear and critical thinking on the concepts that we employ. In our case, in respect to gene drive technologies, that means being clear on the ethical categories, the ethical frameworks through which we're addressing the issue. For example, what is the ethical status of the insect species being targeted by this new technology? What do we really make of the moral status of an individual mosquito or screwworm? Can we really ascribe moral significance to that individual? Well, if not, then maybe we can to the species as a whole. These are the questions that environmental and ecological ethics can help us frame. Or again, what about the environmental impacts of such a technology? We understand that a gene drive release will impact not only its local region, but risks spilling out beyond that region, impacting a neighbour species and having a holistic effect on the ecosystem itself. We need ways of thinking clearly, of measuring in a quantitative way what those impacts might be. Now, there are ways of building in safeguards to the technology itself, but at the same time the scientists themselves are inviting ethicists, philosophers and even theologians to think about those issues critically.
The people who are particularly affected by the screwworm and its devastating impacts are those living in human communities, and especially indigenous communities in some of the poorest regions of the world. And what's interesting about the indigenous communities is they are also very respectful of ecology and their animals and their other creatures. And so the loss of these creatures for them is devastating. It's like losing members of their own family. So it's really important to engage people locally and find out their perspectives before developing these techniques.
There are three main groups we're seeking to work with in this project. First of all, scientists themselves in their laboratories working on the technology. Second, the local communities that will be affected potentially by gene drive releases. And third, those who are working at a very high level in international governance and policy regulatory bodies.
The gene drive research field is very broad. There are some groups that are much further ahead towards application. My specific research is much more on the theoretical side and the enabling technology side. But throughout my work in this field, I have interacted with social scientists, and I found that very useful, both as sort of mentors and as informing how we do research in the lab and what research we do in the lab.
The Laudato Si Research Institute is ideally placed to explore questions like the gene drive technology. This is because it's based at Campion Hall, University of Oxford, where we try and explore the idea of integral ecology in a more holistic way. So we explore, on the one hand, the quiet voices of the marginalized, the poor, but we also bring that knowledge into the policy making and scientific endeavors. So we try to explore biotechnology for a more holistic way.
We're engaging widely with a cohort of scientists, ethicists and others, but we're keen to hear your voice too, and to have your engagement wherever possible. We'll be hosting all these items and more on the Laudato Si Research Institute website, so please do check in and stay in touch with us there.